Kiho's been running our Bern laboratory, basic science laboratory, for 20 years, and PhD. Also was a dentist back in Korea. Came over and has done a great job with research and's got new concepts. Done excellent papers on. He's actually a virologist and retroviruses, and today he's going to talk about adequacy of critical assumptions, understanding and studying biology. I can't wait to hear what this is about. So okay. Go ahead, Kiho.
Thank you, Dave. So during the course of the genetic diversity of humans, mice, and other species, to understand why there are differences in stress response, we collected some data which do not go very well with current assumptions in biological sciences. So since last fall, I started assembling those data sets which are contradicting with current assumptions in biological science. So on my title, I said it's a preview. That means I'm just showing the first two assumptions in biological science which may not be sustainable. And then as I compile more assumptions to challenge, then as I collect them, I like to report those challenges to this group down the road. So our research team at the Bern division, we like to see the human genome as a 4 to the nth system, meaning that when there are 3 billion nucleotides in human genome, we see that as an information system. There's a 4 to the 3 billions information system. Computers, smartphones, we can say 2 to the nth system. So when we are looking at 4 to the 3 billions information, and when we only see about 7.5 billion people on Earth, and those 7.5 billion people share similar frames, that means the genome is highly, highly organized. It's extremely programmed because only 7.5 billion frames are selected out of 4 to the 3 billions. I don't believe there's any computer which can record 4 to the 3 billions as a number. So we like to believe that every nucleotide and its position information matter. But currently, in most of the biological sciences, that's not the case. When we are looking at the genome information system, even though NIH and other scientists claim that they can sequence whole genome in one afternoon, but so far NIH was not able to finish a single chromosome yet. So that means we really don't know the size of the human genome yet. So when we are looking at the human genome, about 1.2% represent the exons or exome as a whole. And the majority of our genome are represented by repetitive elements. Some are transposable, the others are non-transposable. So I would call it about 98% of our genome as a non-gene element. And transposable repetitive elements represent at least 45% of our genome. And one example of transposable repetitive element family is human endogenous retroviruses. So I summarize where we are in terms of current biological research direction. First, the study subject of diverse species, humans, mice, and other animals and plants, are complexly programmed, and their systems are really, really difficult to decode. When you imagine that we are 4 to the nth system, we are not that easy to decode. However, rather simplified, uninformed, and or clonal study designs that includes relevant analytical tools are commonly implemented in these biological studies. So here's the question. Are the current assumptions, which are the foundation of biological studies, sound enough? So there's a very tall building here. There should be a decent foundation so that that tall building can stay there for a long time. So this foundation can be assumptions for biological studies. What if there are some problems with those assumptions? Then that tall building may not stand straight. It will be tilted. But the problem with the tilting tall building is not just a problem of that building. If that building tilts toward the right side here, then the surroundings or neighbors will be affected. So I'm going to challenge two assumptions in biological sciences. So this is the first one. So evolution from a single origin of species by random mutation. In 1859, Charles Darwin published this book on the origin of species by means of natural selection or the preservation of favored races in the struggle for life. The snapshot in the middle is from National Human Genome Research Institute. If you look at highlight here, it says human, mouse, and other mammals shared a common ancestor approximately 80 million years ago. Therefore, the genomes of all mammals are comparably similar. So here's the assumption that I'm going to challenge it. The first assumption to challenge, that is formation of pleiotrophic life forms from a single origin of species through accumulation of random mutation during natural selection. Is this assumption reasonably correct? So this is the current assumption about single origin of species. As you can see here, there's a single origin of species. Then we all share the common ancestor. And random mutation over time will make mouse, human, monkey, dog, chicken, frog, and plant. So with the single origin of species idea plus random mutational event, that means we came from same genome. We learned that mouse and human shares about 85% of conventional gene sequences. But I'd like to remind you that conventional genes only represent about 1.2% of our genome. So if you're only focusing on conventional genes, that's 1.2% of our genome, this single origin of species makes sense because mouse and human share, for instance, ribosomal RNA gene sequence. And also even bacteria have ribosomal RNA gene sequence. And ribosomal RNA gene sequence are highly conserved among all species. That makes sense because maybe we came from a single cell similar to bacteria all together. So when you are looking at the conventional gene codes only, the single origin of species makes sense. But assuming that ribosomal RNA gene is about 1,000 nucleotides. So out of 3 billion nucleotides, we are only looking at the information for 4 to the 1,000s. I mean, 4 to the 1,000s is a big number, but obviously there are bigger numbers. So now we can ask whether this 4 to the 1,000 or the conventional gene information, is it sufficient to compute meaningful distance and lineage among many, many different species? But I'd like to remind you that every nucleotide and its position information matter. So during the course of our study to understand human diversity, we collected some interesting data. We call them the fractals in the genome. So our initial question was, is genome ordered or just random? I shouldn't say random, but genome is only for making conventional genes, or the entire genome information system is ordered into a certain configuration. So we adopted self-alignment and dot matrix presentation to see how genome is organized with repetitive element and others. And we found interesting structures, and we call them RE arrays. I presented this in this group before. So this is from human genome. It's highly organized. It cannot be, you cannot generate this structure randomly. This is another really complex RE array or fractals, highly organized. You cannot generate this in a random. So we did the same with the mouse RE arrays. And this is one example, another highly organized. So we surveyed the entire reference mouse and human genomes, and we compared RE array patterns between these two species. Even though mouse and human share about 85% conventional gene sequences, mouse and human did not share any of these repetitive element arrays or RE arrays. So when you are looking at the entire genome, the majority of genome between mouse and human, they do not share in terms of the structure and sequence. So now we are going back to the question. So if you focus on the conventional gene, like a ribosomal RNA gene, and for instance, if you are looking at 4 to the 1,000th information, the single origin of species makes sense. But now, when we are looking at these RE array data, which are species specific, and that is a lot of data, that means we can say that this single gene or gene-based evolutionary theory or fact cannot stand. So, or we can say that 4 to the 1,000th or conventional gene-based information is not sufficient to come up with a meaningful data for distance and lineage of multiple species. So when you are looking at the rest of, let's say about 60% of our genome is repetitive elements. Now, instead of 4 to the 1,000th, we are talking about 4 to the 1.8 billionth information. 4 to the 1,000th was a big number, but 4 to the 1.8 billionth is a lot bigger number. When you are looking at that level of information, we are completely different. We meaning mouse and human, we are completely different. If you are looking at really small information, like ribosomal RNA gene information, mouse and human, and all the other species, they are similar. Therefore, we can compute the evolution as a single origin of species. However, if you look at the genome information system as a whole, when we are looking at the difference in 4 to the 1.8 billionth information, then it is mathematically impossible for mouse and human to share the same origin. So on the left, there is one human RE array. On the right, there is a mouse RE array. So the single origin of species, we can say that from the genome of common ancestor through random mutation, it will generate new species. So I'd like to ask you a question. Is it really possible to come up with this sophisticated structure or configuration with the accumulation of random events? So random doesn't go very well with this highly ordered structure, because let's say you start from this bottom, and then there'll be random mutations over time. Is it possible to design this unique for human? And is it possible to design this unique for mouse? Because random is random, unless we redefine randomness. So instead of starting from the same or common ancestor for all species, I would say this is a possibility. So instead of starting from the common ancestor, random mutation, maybe there is a genome of human ancestor, and there will be a genome of a mouse ancestor. from here, there'll be random mutations over time. So the caveman's RE array might be quite different from RE arrays of modern people. Same here. There will be genome of mouse ancestor, and then there will be random mutations, and then they'll evolve into modern mouse strains. So based on these findings, we like to propose that instead of all species coming from a single origin, there is a possibility that there are multiple origins of species. Each species has their own ancestor. From there are random mutations over time, and then they evolve. And then there will be natural selection during that process. Again, I like to point out that the information about 4 to the 1.8 billions information is so different, for instance, between mouse and human. That means it is mathematically impossible for mouse and human to share the same ancestor. I'd like to move to the next assumptions. So this is about gene function studies using natural mutant and genetically modified. Genetically modified meaning that it can be transgenic mice, knockout mice, and the CRISPR technologies. So CRISPR is clustered regularly interspaced short palindromic repeats. So the snapshot in the middle is from, again, National Institute of Health. This is about knockout mouse phenotype program. It's called COMP2. So they promoted the knockout mouse phenotyping program as an international effort in mouse phenotyping. And also, they promoted knockout mouse phenotyping program can help precision medicine. So here's the assumption for these gene function studies using mouse strains. That is, gene studies by phenotypic comparison between a pair of mutant modified and its wild-type mouse strains, which are genetically identical. It is very important. They claim genetically identical, except for the gene locus. So I'd like to discuss whether this assumption is sound or strong enough to support all those mutant and knockout mouse studies. So since we are studying the effect of the infection in terms of regulation of signaling pathways and other aspects of the stress response, we introduced TLR4 mutant for our study. TLR4 is a toll-like receptor 4, and it is a key molecule for lipopolysaccharide-mediated innate immunity. So as you can see that, on the illustration, the red we symbolize as a TLR4, which is a receptor for LPS. So for this study, there is a natural mutant. These are not knockout or CRISPR mice. These are C3HEJ is a natural TLR4 mutant. There's a point mutation in TLR4 gene. Therefore, it doesn't make functional TLR4. And Jackson Lab claims that C3HEOUJ is identical to C3HEJ except for that TLR4 locus. So we used HEOUJ as a control to study the effect of TLR4 on LPS stress using HEJ strain. As you can see here, if you look, so we did genomic landscaping to see whether these two strains have identical genome or not. So it was really simple one experiment, but as you can see here, I zoomed in some area on the top gel picture on the bottom. So if you focus on those green highlights, you will see that HEJ is different from HEOUJ in that specific locus. This locus has nothing to do with TLR4. And we only apply one probe out of many probes that we have for this genomic landscaping. This data shows us that HEJ does not have same genetic background as HEOUJ in addition to TLR4 mutation. So we moved further to confirm whether HEJ and OUJ have identical genetic background. So in general, in science, they are talking about transcribed non-coding genes. So this pie chart is coming from one of the Nature review, and we modified the pie chart, but the data is there. The green indicates protein coding regions, conventional gene coding regions. Red indicates transcribed non-coding. That's not what I'm talking about. They are talking about transcribed non-coding. And blue indicates untranscribed. It's not my data, it's their data. However, I like to challenge this pie chart published in Nature Review because in this red area, yes, there are a lot of transcription, and they are saying transcribed non-coding. That is not true. There are a lot of these transcripts which can make functional proteins. They didn't pay much attention. They just disregard them as a non-coding. However, we confirm that some of these produce functional proteins. So in these HEJ and OUJ strains, there are two main types of endogenous retrovirus. I mentioned that endogenous retrovirus is a key family of transposable repair development. One is mutant leukemia virus type endogenous retrovirus. The other is mouse memory tumor virus type endogenous retrovirus. So MMTV ERV produce nice open reading frame, meaning that it produces protein called superantigen. The conventional antigen can only stimulate very small population of T cells. However, superantigen can interact just based on the V beta isotypes. So that means superantigen can stimulate large population of T cells. And there are different isoforms of superantigens. And different isoforms of superantigen can stimulate different population of T cells through V beta specificity. So when that means when different mouse strains have different superantigen profile in their genome, because these are endogenous virus, they are there already. These are not infectious virus, they are endogenous virus. So when different mouse strains have different superantigen profiles, that means they have different immune background. But I'd like to remind you that HEJ and OUJ strain was used for TLR4 study. That's LPS response, and it is directly linked to innate immunity. So basically, HEJ and OUJ were used to study innate immunity through LPS signaling. So we surveyed 56 different mouse strains in terms of their MMTV superantigen profiles. As you can see here with all different colors, it shows the polymorphisms in superantigen in different strains, suggesting that these different strains will have different immune background because superantigen stimulate T cells, and then those T cells, some of them will go to energy state, and then some of them will be deleted. That means different mouse strains have different T cell profiles in their immune system. So this data shows HEJ and OUJ, how they are different in terms of MMTV superantigen profile in their genome. The C-terminus of superantigen decide the V beta or T cell specificity, and it shows the C-terminus of these superantigens. As you can see here, HEJ and OUJ have quite different superantigen profiles. So that means HEJ and OUJ should have different immune background. Then now we can ask, are there differential systemic events of T cell activation, energy, and deletion between C3H-HEJ and HEOUJ strains? That's very possible. If so, are there innately different systemic immune profiles as well as stress responses? In this case, LPS response between the two strains. So that means the data they collected by comparing HEJ and OUJ in terms of LPS signaling pathway or innate immunity. Is that just because of TLR4 mutation or the main differences in their immune system? It will be very important to find out which is the case. So now I'm getting into another type of mouse research. It is knockout system. So we introduced CD14 knockout mice in combination with TLR4 mutant to study LPS signaling. So it briefly describes how this CD14 knockout is made. There's CD14 knockout construct, and then it will be electroporated into 129 derived embryonic stem cells. And then those recombinant stem cells will be injected into C57 black 6J derived blastocyst. There will be a pseudopregnant mice, and then there will be chimeras of 129 and C57 black 6J. And then they claim that about 10 generations of backcrossing with black 6J will make the CD14 knockout strain as a co-isogenic with black 6J. This is very important, co-isogenic with black 6J, so that we can use black 6J, which is a backcross strain for CD14 knockout, as a wild-type control to study CD14 knockout. So here is the definition of the co-isogenic strains. Co-isogenic strains are one type of inbred strain that differs by a mutation at a single locus, and all of the other loci are identical. All of the other loci should be identical. Then we can call them as a co-isogenic, and then we can use them as their control. So this is same signaling pathway illustration. So LPS has TLR4 as a main receptor, and also CD14 is another important receptor or co-receptor for LPS. So that's the reason why we introduced CD14 knockout for our studies. But using the same genomic landscaping technologies, as you can see here, CD14 and its backcross control strain do not have identical genetic background. It's so obvious that they do not have identical genetic background. And in addition to this, we found some more interesting data, and we dissected further. So we just happened to find this. So one of our research interests is understanding the roles of glucocorticoid receptor in stress response in different human population, and also different isoforms of glucocorticoid receptor. So glucocorticoids are involved in growth, development, stress response, and maintenance of homeostasis, among others. And this is a transcription factor, and human glucocorticoid receptor gene is on chromosome 5. And I'm not going to explain HPA axis into this group. So we were interested in how CD14 knockout affects the expression of glucocorticoid receptor. So we found something interesting, that is there's a decrease in glucocorticoid receptor expression at earlier time for black 6J and CD14 knockout. However, that decrease in expression was prolonged until day 1 in CD14 knockout, but not in control strain. this is a significant difference here. But we noticed that something interesting. That is, if you look these two here, there's kind of shift in terms of the size. And also, it was repeated here. So we assume that what is going on? Did we make any mistake with the Western blot? So we did all the check. We couldn't find any mistake. So we decided to look at what is going on with the cDNA. So we confirmed that glucocorticoid receptor from Black6J is different from glucocorticoid receptor from CD14 knockout. I'd like to remind you that this CD14 knockout should be different only at one locus, that is CD14. But now we are confirming that CD14 knockout and Black6J is control have different glucocorticoid receptor. And glucocorticoid receptor is very, very important gene or protein in stress response. And also we independently confirmed that yes, glucocorticoid receptor, size of glucocorticoid receptor in terms of the protein, they are different. We independently confirmed that. And we tested the function of these two different glucocorticoid receptor isoforms, one in CD14, the other one in Black6J. Obviously, they have different transactivation potentials. That means they have different protein size, cDNA, and function. So in addition to the difference in the CD14 locus, now this CD14 knockout mice have different glucocorticoid receptor gene. So now we came up with this question. Maybe the genetic background of CD14 knockout mice might be quite different from what we expected. So see, when there was a 10 generation of backcrossing, there will be CD14 knockout strain. They claim that this CD14 knockout is co-isogenic with C57. Therefore, we can use them as a control. But we decided maybe not, because we already showed that they have different glucocorticoid, more than one locus. So we decided to look at the genes surrounding this CD14 locus to see whether glucocorticoid receptor is right next to each other so that they move together during the crossing over event. So we mapped them on chromosome 18, and then now as you can see here, glucocorticoid receptor is right next meaning that they're about more than 2 million nucleotides away, but when you are looking at 3 billion, I can call it right next to each other. So CD14 and glucocorticoid receptor, they are neighbors. So likely, when there's a crossing over, I guess that chunk of DNA or chromosome is moving together. You cannot separate them out. They move together. Unless you confirm that all mouse strains share 99.9% of genome, there's no way that you can separate these out. So we surveyed what genes are around that area. So you can see the CD14 and glucocorticoid receptor on this side. From here to this, that's about 2.7 million nucleotides. And we look at 2 million upstream and 2 million downstream. We survey different genes annotated at NCBI. You can see here that in this region, between CD14 and glucocorticoid receptor, we have a very important gene family, protokaryoterin. I'm not an expert in neuroscience, but I can say that protokaryoterin family is a key protein of neuronal network in the brain. Some people say it decides the blueprint of the brain. And those protokaryoterin family were in between CD14 and glucocorticoid receptor. If the glucocorticoid receptor was co-segregating with CD14, then we can say that there's a good chance that protokaryoterin will co-segregate with all together. So now we dissected further to confirm whether they are segregating together. As you can see here, we decided to do the genotyping for these glucocorticoid receptor. We have Black6J. We found that they have 17 CAG repeat. It converts to Q here. And we have black 6J-F1 and 129F1. 129 embryonic stem cell was used to make CD14 knockout. That's why we have 129 strain, and we have CD14. So as you can see here, black 6J has 17 Qs, but this 129 has only 8, and CD14 has only 8 here. So that means CD14 knockout is supposed to be co-isogenic to black 6J. But however, when we are looking at glucocorticoid receptor gene, it goes with 129 instead of black 6J. And it clearly shows that glucocorticoid receptor is co-segregating with CD14 knockout locus. So we analyze what genes are in between CD14 and glucocorticoid receptor. So we picked some of those genes to see whether those genes are co-segregating with CD14 and glucocorticoid receptor into CD14 knockout mice. So we selected these 4 genes. So at DNA level, we were able to confirm that yes, the CD14 knockout goes with 129, meaning that it has a 129 PCDH beta 5 instead of black 6J's gene. So this data shows that CD14 has 129 gene profiles in this area instead of black 6J. And also we were able to confirm at the protein level for the other genes as well. And it clearly shows that the area between CD14 and glucocorticoid receptor on chromosome 18 of 129 strain co-segregated into the background of black 6J. So that means clearly black 6J is not a co-isogenic strain for CD14 knockout mice. Therefore, black 6J cannot be used as a control for CD14 studies using that knockout strain. So this is a summary of the findings from knockout studies. So the co-segregation of knockout locus might be quite common in all knockout strains because you cannot control crossing over event. We still don't understand how crossing over happens. That means we cannot control them. Unless mouse strains all share 99.9% of genome sequences, now we have a problem here. But as I showed you before, mouse strains do not share 99.9% of genome sequences. So the co-segregation of knockout locus is quite common, not just for the CD14 knockout strain. Then the scientists claim that 10 times of backcrossing is sufficient to generate a co-isogenic strain. But nobody confirmed that these are co-isogenic strains in any studies. They only assumed what they guessed. They claimed, but nobody confirmed that they have an isogenic strain, a co-isogenic strains, meaning that 2 strains are different at only 1 locus, the rest are identical. Nobody confirmed, but everybody, I shouldn't say everybody, almost everybody believes that is the case. If you ask me, how do you think? Then I would say that's not sufficient. And I'm not even sure whether we can make a co-isogenic strain down the road. And what are the effects of polymorphic segments inherited from the ES cells, like 129 in CD14 knockout, on phenotypes of knockout strain? Nobody studied and nobody confirmed. So when we are looking at CD14 knockout studies, then we attribute all the difference between these 2 strains, knockout and black 6J, as the function of CD14. But now you saw that there are many different isoforms of genes, not just CD14, glucocorticoid receptor, protocaryoterin, and many other genes. How do you know that's the function of CD14? So for me, it makes sense to confirm a pair of knockout and wild-type control strain share the same genetic background except for the knockout locus. I mean, it's a common sense to compare. Do they have really identical genetic background? If not, then we need to discuss. So obviously, mouse strains do not share 99.9% of genome sequences. And also, there is no single mouse genome completely sequenced yet. There's no single mouse genome completely sequenced. Even NIH's reference genome is not completed yet. And even worse, it seems it's really difficult to estimate the size of individual chromosomes in mice and humans. And I showed this data in my previous presentation here. So mouse chromosome Y was annotated as 16 million nucleotide until 2013. 16 is 1/6. But in 2013, NIH quietly updated the mouse chromosome Y. Now it is 92 million nucleotide. So almost 6 times larger. That means NIH is having really hard time to estimate the size of individual chromosomes, not to mention the sequencing entire genome. So in many mutant and genetically modified modified gene mouse studies, we see the phenotype of knockout or mutant. So currently on the top, we assume, what they assume, current gene study designs is based on identical genomes assumption. Except for the target locus, the rest are identical. That's why they are claimed as a co-isogenic. However, from our study, we confirmed that that's not the case. They have different genomic background. And also, another thing is that they did not confirm that they have identical sequences anyhow. So all the phenotype that we reported and published and trying to translate into human medicine, is it because of the knockout of that specific locus, or in combination, or just for the different genomes, just different mice? Is that the phenotypes of different mice, or the phenotype of knocking out that specific gene? So a couple of years ago, there was a paper called Genomic Responses in Mouse Models Poorly Mimic Human Inflammatory Diseases. So this is a kind of summary paper from a big group of scientists who studies inflammation and host response to injury. So basically, they mostly failed with their big study, and then now they are saying that mouse models poorly mimic human inflammatory disease. But I like to say that at least it is due to incompatible genetic backgrounds employed in mutant and modified mouse studies. In other words, those gene-focused studies using mutant and knockout studies, when you are using inadequate controls, the data that you generate from those studies may not be that useful, and you may not be able to translate that into humans. So I like to say that it's not the fault of mice, it's a human fault. So what are the impacts of the difference in genetic background in each pair of mutant, modified, and wild-type strains? We don't know. So in order to accomplish a tangible translation of mouse studies in human medicine or some other species, we need to reevaluate the previous mouse studies, and we need to confirm genetic compatibility for new studies instead of just assuming that they are identical. We need to confirm the genetic compatibility for new studies. But based on my understanding, confirmation of genetic compatibility is a big task to do. So our current biological assumptions sound enough to support meaningful hypothesis, studies, and outcomes. I hope we stay on the left instead on the right. So in the future, I like to give next set of assumptions challenge to this group, and I'm compiling that report at this moment. Thank you. Yes.
Kiho, nice work. Your assumption that you start off with one organism and branch out with the Darwin theory has been also challenged by author Stephen J. Gould, who wrote a book called A Wonderful Life, about fossils from virgin shale up in Canada. He basically showed that if there was no competition, you had all different kinds of life that was multivariate in them. They kind of grew and started competing, and we narrowed down to specific species. So that's his theory that when there's no competition, you have all different kinds of looking life, and then eventually when competition got up, then fewer and fewer species evolved, so the tree's upside down according to this theory. I looked through Nature. They give news every day, and they talked yesterday about a ultra-conserved non-coding region of the genome, which is really enhancers, not so the genome itself isn't the part that's conserved, but there may be parts of our DNA that's ultra-conserved, but that serves as either enhancing or promoting or down-regulating genes. So there may be, as you say, the junk DNA is really has more function than life.
So I hope maybe within a couple of 100 years, junk DNAs have a new life. That would be great.
What's the role of mice caught in the wild in translational studies that will be more applicable to what humans behave rather than using laboratory-generated mice?
Yes, so wild caught, I know one scientist, Dr. Maury Gardner, he used to go out to LA area and he caught wild mice for the studies. Actually, I used one of his mouse strain from the wild for MMTV study. So in typical mouse studies, so we have inbred strains and also like ICR outbred stock. So sometimes, yeah, outbred stock might be a lot better than using inbred strain because it mimics the human population. I mean, it's not really wild, but outbred stock is that makes more sense to use.
There have recently been some human trials using CRISPR technology to edit the genome of grown organisms. Is that something you see potentially playing a role in some studies to have essentially isogenic strains or very inbred strains that have not undergone germline mutations to potentially edit a very specific site in an adult organism?
Are you talking about humans or mice?
In mice now, or in lab studies, but we're doing some in humans. Does that have a role in that?
So the CRISPR is on my list of challenges. The CRISPR, they need to resolve their off-target effects. Since we don't know, there is no reference genome, so that means they really don't know what they are targeting. I mean, obviously, they are targeting gene A, but gene A will have multiple pseudogenes, for instance. So same construct will target multiple loci, but currently, they don't have that ability to precisely know where they are targeting. So in some way, before moving forward with CRISPR technology, they should sharpen their technology so that they really know what they are targeting. Otherwise, down the road, there might be some new scientists like me saying that all the CRISPR studies, we don't know what is happening. It's like similar to CD14. They thought CD14 knockout is only different with Black6J, only with CD14, but now we are showing that they are different in many, many loci. We only look at that 2 million nucleotides out of close to 3 billion. If you look at the entire genome of CD14, we'll see many, many differences. Basically, these are different strains. They are not co-isolating. So CRISPR may face the same challenge down the road. Yes.
Very nice, Kiho. Fascinating. Dave Greenhall asked the question in a much more sophisticated way than I can, but if we go back to the first part of your talk, that there's not a single origin of the species. this non-coding region, this huge part of the DNA, this non-coding, highly organized in different ways between the species, must have a very definite effect on what happens.
Yes.
As to how we look.
Yes.
I have to go lie down.
Kiho, that was just great. I particularly enjoyed the way you blew up multi-billions of dollars worth of historical research based on a fundamentally scientific approach. Most of us in the audience are sitting here predicting that this news is not going to be popular at NIH, which has funded all these studies. And I'm sure you've sort of thought about this, stayed awake at night thinking about it. How do we go forward from here? Do you think we just need to spend a lot of more money to figure out whether our models were a little wrong, or a lot wrong, or completely useless?
So if I'm in charge, let's have that assumption. I'm in charge. Then I would propose to have a dialogue or a task force to discuss or to evaluate our current mouse models or any other animal models, whether our assumptions are strong enough or sound enough. So if I'm in charge, I would do it. But obviously, I'm not in charge. So this is my approach. So I already made a list of assumptions that I'm going to discuss and challenge. So that's why I said, again, preview. I have many other assumptions to challenge. So it will take a few more months to compile them all. So if the department allows me to do so, I like to put that information into department website for the general public and other scientists. If they are interested, they can read. So that's my next step. And also, I'm trying to have some discussion with NIH, mainly like a program officers. So somehow I have some progress with that approach. So with NIAID, National Institute of Allergy and Infectious Disease, they have a radiation countermeasures program. Basically, they are getting ready for nuclear war or nuclear accident. And they are interested in setting up the triaging system using biodosimetry. So I am in contact with the NIH's radiation countermeasure program. I'm bypassing study sections, but I'm talking to NIH. So that's my another approach, just to talk to the government officers, so that. But I know there is a long way to go, and also I was quite hesitant to prepare this presentation and present, but about 2 months ago I decided to go ahead, so I cannot stop.
We thank you. We're the richer for it.
Thank you. Yes.
Very, very provocative. I'm just wondering, your first slide where you talked about the foundation and building this straight and upright, and how you're challenging basic assumptions, and it could have collateral damage if that foundation is faulty. You're thinking at a different level than some of us. Can you expand on that? What do you think is the effect? What is the damage that you're talking about when you're building into both?
So actually, for this presentation, I listed 3 assumptions to challenge, but I realized that my slides are growing further and further, so I decided to cut number 3. So that is about genetic identity. So one damage to the society with this wrong assumption is that current DNA identification system for criminal justice and also identification of mice, cell line, or seed, or anything, it is based on looking at short tandem repeat, about 20 of them. But they have 2 key assumptions. One is everybody in one species, they share 99.9% of genome sequences. And also, the second assumption is that we have completely sequenced reference genome. But obviously, those two assumptions are false. So that means we might be putting wrong people into the jail. Or we might be taking wrong people out of the jail, like an innocent project. Sometimes they take people out from the jail based on the DNA evidence. I'm not sure whether that is right or not. So that is one. And also, by spending all these money for mouse studies, for instance, and what if all the data that we generate are from the bad assumptions? And now a lot of scientists and institutions are trying to translate those findings from animal studies into human medicine without knowing that there are some problems with their assumptions. Then we may spend next couple of 100 years based on those wrong assumptions. And that's a huge damage to the society. It's not just for the papers published by the scientists. It might be relatively minor because they are well confined. We know that they are published here. But by tilting these scientific findings based on wrong assumption, then the impact to the society will be quite diverse. Like DNA identification and advancement of medicine will be delayed. So the damage will be amplified from there. And also, what if our future scientists will use current publication as their reference to design their future studies? It will only amplify and waste.
Kiho, I think it's very easy to follow what you say because it makes a lot of sense, and I agree with you. The math just simply doesn't support that this is all by chance. The question I sort of kept having in my mind as this presentation warmed up this week, what can we do today? And one of the examples from clinical medicine is there's a lot of things that take a lot of rigor to identify. That we start with screening tests, right? So we screen a whole bunch of people for one thing, and we figure out, okay, who's at risk and so forth. Is there even an opportunity for this as an example? You looked at your CD14 knockouts. Is there a way to just sort of say, okay, you know what, I'm going to ignore all the multitude of differences. If this is a knockout, this is a big deal. We test it on a big level. If there's no difference, then we move on. But if there's a difference, then we look and see, how appropriate the model is. Because I would imagine that the rigor of really identifying that your study subjects are isogenic or isogenic, unless you remember the term that you
Co-isogenic.
is very, very, very rigorous to do ahead of every single study. So how would you see that occurring so that you don't waste a lot of time doing that just to find that there's no useful finding of a study? Is there some role for screening?
Yes, so this is what I would propose if NIH asks for that problem. So let's say CD14 knockout and controls. Then before we decide that this is a co-isogenic strains, then we should look at the genetic backgrounds at high level. And we should have a kind of advisory group and evaluate the data. And then we confirm that, yes, this is 99.9% identical. So therefore, we define this as a co-isogenic. So there should be a group of, or an institution which confirms that these are co-isogenic instead of randomly. For instance, Jackson Lab declares that these are co-isogenic, but after all, they are not co-isogenic. So there should be a government institution confirming all these different co-isogenic strains, and then give an approval, like they are 98% identical. And they can go further. They can study the genetic background further and provide more information in terms of where they are different. Because I don't believe that there will be a 100% co-isogenic strain. But at least there should be a government institution confirming the genetics of co-isogenic strains. That might be the only way to go forward.